In this question, we're asked to estimate the pH and hydronium ion concentration of a solution using pH indicators. In chemical analysis of separate samples of an unknown solution, phenolphthalein was colorless, bromothymol blue was blue, and phenol red was red. What is the estimated pH and hydronium ion concentration of this solution? So in order to do this, we are going to need our data books. And what we're going to look for is we are going to look for each of these acid-base indicators and use that information to determine what the color range would have to be. So our first one is phenolphthalein. So with phenolphthalein, we are told that the color is actually colorless. When phenolphthalein is colorless, what that means is that it has a pH of less than or equal to 8.2. Next, we are told that bromothymol blue is blue. What that means then, when bromothymol blue, is that the pH has to be equal or greater than to 7.6. And finally, we are also told that phenol red has a red color. This then means that it has a pH equal or greater than 8.0. So what we end up seeing here is we can actually make a little bit of a number scale. We know that for phenolphthalein, it has to have a pH of equal to or less than 8.2. For the bromothymol blue, it has to be 7.6 or greater. And for the phenol red, it has to be 8.0 or greater. Where all of these overlap is the range that our solution's pH has to be in. And what we see is they all overlap between 8.0 and 8.2, this range here. Now, we can't be certain exactly where it falls within that range. However, given that it's a fairly small range, we can estimate that the pH is going to be about equal to 8.1. It's not perfect. It could be anywhere between 8 and 8.2, but since it's a small range, we'll just estimate that it's in the middle of that range. And then for the concentration of the hydronium ions, that's going to be equal to 10 to the negative pH. So 10 to the negative 8.1. We only have one significant digit in that pH, so the concentration would approximately be, because we are estimating the pH in the first place, the concentration would be approximately 8 times 10 to the negative 9 moles per liter.